Okay, let's unpack this. Imagine your DNA is, well, it's like a massive multi-volume set of encyclopedias.
Right, just an overwhelming amount of information.
Exactly. We are talking millions and millions of letters making up the ultimate instruction manual for your body. And, if you were to go in and change a single B to an F in one of those volumes, no big deal, right?
You wouldn't even notice it.
Yeah, just a tiny typo. You'd think the body would just read past it. But in the human body, that single typo can cause a total catastrophic systemic collapse.
It really can.
So today we are opening up that manuscript for you, the listener, to decode the hidden instruction manual of our bodies. We're looking at how this code works, how traits are handed down, and, well, the profound, sometimes really heavy choices modern medicine offers us when that code has a misprint.
It truly is the most complex manuscript in existence. And what's fascinating here is that understanding genetics isn't just, an exercise in biology. It is fundamentally about understanding the blueprint of human variation. It's our survival mechanisms and that delicate chemical balance that keeps us alive minute by minute.
Yeah, it's everything.
It is. But to understand the whole human being, we have to start down at the microscopic level, right at the actual code, before we can even see how a misprint affects the entire organism.
Yeah, the misprints, we usually call these mutations, which I know it always sounds like a sci-fi movie trope.
Right, like you're going to get superpowers.
Exactly. But it's really just a permanent change in the DNA. And these usually happen when cells are dividing, right? Like the biological machinery is essentially photocopying millions of pages, and occasionally it just blinks and makes an error.
Precisely. DNA replication is incredibly accurate, but it's definitely not infallible.
Right.
And there are mutagens in our environment that can aggressively increase the rate of these errors. Things like X-rays, ionizing radiation, or specific chemicals that literally physically damage the DNA strand.
Oh, wow. So it's not always just a random mistake?
No, but often it is just a spontaneous copying error. Sometimes we see large-scale chromosomal mutations where huge chunks of genes get rearranged, or even whole chromosome mutations.
Like what?
Well, a well-known example is Down syndrome, where an individual inherits an entire third copy of chromosome 21 instead of the usual pair.
Okay, yeah. But the ones that really blow my mind are the point mutations. We're back to that encyclopedia analogy. A point mutation is literally changing one single nucleotide base.
Just one letter.
Yeah, you have substitution, where one letter is swapped, deletion, where a letter is dropped completely, or insertion, where an extra one is squeezed in.
Right. It alters the sequence.
It's like changing one letter in a recipe. If the recipe says bake the cake and a substitution changes the B to an F, now it says fake the cake, the instruction is entirely ruined.
That's a great way to look at it.
But I really want to understand how that microscopic change scales up. Take sickle cell disease. I know it stems from a point mutation, but how does changing one single letter actually reshape a red blood cell and cause such a massive crisis?
It is honestly one of the clearest, most sobering examples of molecular causality. In sickle cell disease, you have a single substitution point mutation on the gene that codes for hemoglobin.
Okay.
There is a specific sequence of DNA that should read GAG. But because of a copying error, it mutates to read GTG.
So just an A swapped for a T?
Exactly. An A is swapped for a T, and that is the entire genetic origin of the disease.
So the cell reads GTG instead. What does that actually do to the physical materials in the blood?
Well, proteins are made of chains of amino acids, and the DNA sequence tells the cell which amino acid comes next. Because of that one swapped letter, the cellular machinery removes an amino acid called glutamic acid.
Okay.
And it inserts a completely different one called valine. Glutamic acid has a specific electrical charge and shape. Valine behaves very, very differently.
So the building block is just wrong.
Right. That single substitution fundamentally alters the physical structure of the hemoglobin molecule, which is, the protein responsible for carrying oxygen inside your red blood cells.
And hemoglobin normally just floats around nicely inside the cell, right?
Normally, yes. But this mutant hemoglobin, because of the valine, it tends to stick to other hemoglobin molecules when oxygen levels drop. They polymerize.
Well, they stick together.
Yeah, they form these long, rigid, microscopic rods inside the red blood cell.
That's incredible. So it's essentially building scaffolding inside the cell where there shouldn't be any.
And that scaffolding forces the entire red blood cell to collapse. It goes from its normal, flexible, donut-like shape into a rigid crescent or sickle shape.
Hence the name.
Exactly. Now, normal red blood cells bend and squeeze through the tiniest capillaries in your body. Sickled cells are stiff. They act like a log jam.
Or they get stuck.
They get stuck in those tiny blood vessels, entirely blocking blood flow to organs and tissues. That oxygen starvation is what causes the severe, agonizing pain crises and life-threatening organ damage associated with the disease.
And that all cascades from one letter swapping an A for a T.
It's staggering.
But I have to push back on this premise. If changing one letter can cause something as painful, dangerous, and biologically expensive as sickle cell disease, why hasn't nature just edited mutations out entirely?
It's a fair question.
Right, like, if mutations cause diseases, aren't they inherently bad for a species' survival?
What's fascinating here is that mutations are actually the foundational engine of evolution. Without them, there is absolutely no genetic variation.
Wait, really? None?
None. If DNA replicated perfectly every single time without a single error since the dawn of life on Earth, life would never have adapted. We would still be single-celled organisms floating in a primordial soup.
Oh, wow.
The errors are what give a species a menu of new traits to survive changing environments.
So it's a numbers game. The mistakes are actually a necessary feature, not a bug.
Absolutely. And it's vital to realize that the vast majority of mutations do not cause disaster. Most are completely neutral.
Because they don't do anything.
Right. A huge portion of our DNA consists of non-coding regions, sections that don't actively build proteins. So a typo there often does nothing at all.
Right.
Furthermore, our genetic code is what biologists call degenerate.
Degenerate? That sounds like the code is morally compromised.
I know, it's a funny term. In biology, it just means there's built-in redundancy. There are multiple different three-letter DNA combinations that code for the exact same amino acid.
Oh, I see.
So you might get a point mutation that changes the third letter of a sequence, but the new sequence still asks the cell for the exact same amino acid as the old sequence.
So the final protein is built perfectly anyway.
Exactly. The body never even knows a mutation occurred. We only see these dramatic systemic effects when a mutation happens to strike a critical active site of a vital protein, like with the hemoglobin.
Okay, so that perfectly explains how the code mutates and why those variations exist, but I want to explore how those altered genes, or even just normal variations, actually get handed down to you.
The inheritance piece.
Right, the genetic lottery.
Ah.
We all know we get half our chromosomes from our biological mother and half from our biological father. We inherit these matching homologous pairs.
Yes, 23 from each.
And usually we think of it as a tug of war between a dominant allele, which is kind of the bossy one that takes over, and a recessive allele, which hides in the background unless it meets another recessive allele.
That is the classical Mendelian view, yes.
Yeah.
But the inheritance landscape is far more nuanced. It isn't always a case of one allele completely dominating another.
They can share.
Yes. Often, they share the stage through something called codominance.
Right, and a classic example of this is the ABO blood group system. I always found this fascinating. The allele for blood type A and the allele for blood type B are codominant.
Precisely.
If you inherit A from your dad and B from your mom, neither one backs down. Your body simply expresses both of those markers on your red blood cells, and you have AB blood. The O allele is the only one that acts recessively to both of them.
That's spot on.
And what's really amazing is that we humans aren't the only ones operating on the ABO system. Chimpanzees and bonobos have the exact same blood groups, which is just this beautiful, tangible proof of our shared evolutionary timeline.
It is a striking reminder of evolutionary continuity. The blood flowing through our veins operates on the exact same chemical tagging system as our closest primate relatives.
But let me challenge you on how we actually map out these probabilities. In high school biology, everyone learns to draw those little four-box grids, Punnett squares.
Oh, yes, a classroom staple.
You put the parents' genes on the outside, fill in the boxes, and figure out the odds. It makes genetics look like incredibly clean, predictable math. So if two parents are carriers of a recessive trait, the Punnett square tells us there is exactly a 1 in 4 chance their child will express that trait.
On paper, yes.
Does that mean if a couple has exactly 4 kids, one of them is mathematically guaranteed to have it?
If we connect this to the bigger picture, the answer is a resounding no. And that is a massive misconception that even some medical patients struggle with.
It's not a guarantee.
No, Punnett squares illustrate theoretical probability. They do not predict a guaranteed destiny. In the real world, we are dealing with something called sampling error.
So it really is just a coin toss every single time.
Exactly. Every single time a child is conceived, it is an entirely independent genetic event. The biology of the sperm and the egg has no memory of the fertilization that happened 2 years prior.
That makes total sense.
Right. A couple carrying a recessive trait could have 4 children, and none of them inherit the trait. Or They could have 4 children and all 4 express it. Human families are simply too small to reliably reflect those perfect theoretical ratios. The sample size is statistically noisy.
Which explains why geneticists don't usually base their fundamental research on human family trees.
They really can't. The numbers don't work. This is why scientists rely so heavily on experimental organisms like Drosophila, the common fruit fly, or E. coli bacteria.
Because they breed so fast.
Yes. These organisms have incredibly short life cycles, and they produce hundreds or thousands of offspring in a matter of days. When you look at an entire population of 4,000 fruit flies, the actual genetic results will align almost flawlessly with the theoretical math.
The law of large numbers.
Exactly. The laws of probability require volume. When you look at a human family of 3, the math often looks entirely random.
Okay, so we've established that for most of our chromosomes, inheritance is this massive, unpredictable coin toss between matched pairs. But there's a huge exception to these rules, and it happens on the specific chromosomes that determine biological sex.
Yes, the sex chromosomes completely change the arithmetic of inheritance. Humans have 23 pairs of chromosomes. The first 22 pairs are autosomes. They match perfectly, gene for gene.
They've had the same instructions.
Yes, carrying the exact same categories of instructions.
Yeah.
But that 23rd pair is entirely different.
Here's where it gets really interesting. Biological females usually have a matching pair, 2 X chromosomes, but biological males have a mismatched pair, an X and a Y. And if you look at the actual specs on these 2 chromosomes, it is a wild, almost comical mismatch. I like to think of it as a car analogy. The Y chromosome is a stripped-down, ultra-lightweight track car. It has a steering wheel, an engine, and absolutely nothing else.
That's a very apt way to visualize it.
It's tiny. It's about 23 million base pairs and roughly 78 protein-coding genes. Its primary job is basically flipping one switch, the SRY gene, which triggers male development in a fetus.
And that's about it.
Now compare that to the X chromosome, which is a fully loaded family SUV. It is massive. It contains over 150 million base pairs and carries roughly 1,200 genes.
A huge difference.
And these genes aren't just about biological sex. They code for all kinds of essential, everyday human traits like blood clotting and color vision. So my question is, why does this chromosomal mismatch mean that certain genetic diseases hit men so much harder than women?
It comes down to vulnerability and the lack of a biological backup system. Because the X chromosome carries so many vital instructions that the Y chromosome completely lacks, genes located on the X chromosome are called sex-linked.
Okay, sex link.
Let's look at how this plays out. If a female inherits a faulty recessive allele on one of her X chromosomes, say, the allele that causes red-green color blindness, she almost always has a healthy, dominant allele on her second X chromosome.
Oh, I see.
That second, healthy X chromosome effectively masks the faulty one. She becomes a carrier of the trait, but her own vision remains perfectly normal.
So she has the SUV, and she has a backup SUV in the garage, just in case.
Precisely. But biological males, with their XY pairing, do not have a backup. If a male inherits an X chromosome from his mother that carries a faulty recessive gene, he will definitively express that disease or condition.
Because the Y can't help.
Right. His tiny, stripped-down Y chromosome has no corresponding genes to override or mask the error. He is entirely exposed to whatever's written on that single X chromosome.
I remember reading about early experiments with fruit flies that cracked this code, but I never understood exactly how eye color proves sex linkage.
Yeah.
How did scientists actually connect those dots?
It was Thomas Morgan's groundbreaking work in the early 20th century. He was breeding Drosophila and noticed a spontaneous mutation where some flies had white eyes instead of the normal red eyes.
Okay.
As he mapped the inheritance patterns over thousands of flies, he realized something striking. The white eye mutation was showing up almost exclusively in the male flies. The females carried it, but rarely expressed it.
Because they had a backup X.
Exactly. That observational leap, connecting a physical trait to the sex of the organism, proved that certain genes were physically sitting on the sex-determining chromosome.
And in humans, the consequences of this sex linkage range from minor inconveniences to incredibly severe conditions. You mentioned red-green color blindness, which affects roughly 7 to 8% of men globally, but less than 1% of women.
Yes.
But then you have something like hemophilia A.
Hemophilia A is a profound example of the high stakes of having no genetic backup. It is a severe deficiency of a specific blood-clotting protein known as factor VIII.
And the instructions for that are on the X chromosome.
Entirely located on the X chromosome. So males who inherit the faulty allele simply cannot produce the protein. Their blood cannot clot properly. Without modern medical intervention, a minor internal injury, or a simple scrape, could cause fatal bleeding.
That is terrifying.
It is. We treat it today with genetically engineered factor VIII, but it perfectly illustrates how a single missing instruction on a mismatched chromosome can threaten a person's life.
We've seen how sex chromosomes leave men vulnerable to missing genes, but what happens when the chromosomes are perfectly matched autosomes, but they both carry a microscopic error? This brings us to a condition that shows how one broken protein can bring down the whole system, cystic fibrosis.
Cystic fibrosis, or CF, is arguably the most comprehensive example of how a single molecular failure cascades through the entire human body. It is an autosomal recessive condition.
So not on the sex chromosome.
Correct. The most common cause is a specific mutation known as DF508, located on chromosome 7. Because it's recessive and on an autosome, it takes 2 carrier parents for a child to inherit the disease, and it affects males and females equal.
So what exactly is breaking down at the cellular level?
The mutation affects a specialized protein called the CFTR channel. You can think of the CFTR channel as a microscopic, highly regulated doorway built into the membranes of your epithelial cells, the cells that line your organs.
Okay, a doorway.
This doorway has one primary job: to pump chloride ions out of the cell and into the surrounding fluid. But in individuals with CF, that protein doorway is misfolded and faulty. The door is permanently locked shut.
So the chloride ions can't get out.
Right. They cannot exit. They get trapped inside the cell.
I get that the protein fails to pump chloride out, but I struggle with the scale of this. How does a trapped chloride ion, which is essentially just a component of salt, cause someone's lungs and digestive system to fail? That feels like a massive biological leap.
This raises an important question about the hidden physics governing our biology. The leap is entirely driven by a fundamental force: osmosis.
Osmosis, okay.
Normally, when those chloride ions are pumped out of the cell, water naturally follows them across the cell membrane to balance out the concentration. It's just simple physics. That water is absolutely crucial.
Because it hydrates things.
Yes. It mixes with the mucus lining your lungs, your pancreas, and your reproductive organs, keeping that mucus thin, runny, and slippery.
Ah, so if the chloride is trapped inside the cell,
The water stays trapped inside the cell with it. It doesn't flow out to thin the mucus.
Yeah.
As a result, the mucus coating the outside of the cells becomes incredibly dehydrated. It transforms from a slippery protective layer into a thick, sticky, impenetrable sludge.
and that sludge is what sets off the domino effect across the body. Let's walk through that, starting with the lungs. Normally, respiratory mucus is sticky enough to trap inhaled dust and bacteria, but thin enough that microscopic hairs can swip it up and out of your airways to be coughed or swallowed away.
That's the normal process.
But with CF, this dehydrated sludge traps the pathogens and locks them deep inside the lung tissue. The body can't clear it. It leads to chronic, severe bacterial infections and physically blocks the airways from absorbing oxygen.
And the digestive system suffers just as heavily. Your pancreas produces vital enzymes that you need to break down the food you eat.
But the sludge gets in the way there too.
Yes, that same thick mucus physically blocks the pancreatic duct. Those digestive enzymes can never reach the small intestine. A person with CF can eat thousands of calories a day and still suffer from severe malnutrition because their body physically cannot access the nutrients.
Oh.
Even worse, those trapped enzymes can become active inside the pancreas and start digesting the organ itself, destroying the cells that produce insulin and frequently triggering diabetes.
It's an absolute systemic shutdown. It even impacts the reproductive system, physically blocking the cervix in women with thick mucus or completely blocking the sperm ducts in men, making infertility a major symptom.
It really affects every system.
But there is one highly specific, almost poetic symptom that has been noted throughout history regarding the sweat glands.
Yes, the sweat glands. In the sweat glands, the CFTR protein doorway actually works in reverse. It is supposed to reabsorb chloride from your sweat back into the body before the sweat reaches the surface of your skin.
Oh, I didn't know that.
Yeah, but because the doorway is locked, the chloride and the sodium that accompanies it remains in the sweat. Long before we could sequence DNA, CF was recognized by midwives and parents who would kiss a newborn baby's forehead and taste an overwhelming, unusual amount of salt on their skin.
It's a heavy reality, and it's surprisingly common. In the UK alone, about 1 in 25 people carry the faulty recessive allele for CF without ever showing a single symptom. Which brings us to the final frontier of this exploration.
The screening.
Right. When a condition like CF can cause that much systemic damage, it makes sense that medical science wants to see it coming. Let's talk about genetic screening and the ability to look into the future.
The development of genetic screening has been revolutionary. It allows us to peer into the code and anticipate these molecular failures, either right at birth or even before conception.
It's essentially proofreading the manuscript before it goes to the final printer. The most universal form of this is newborn screening. Globally, babies undergo a simple heel prick blood test on their first few days of life to check for various conditions.
Yes, it's very standard now.
And one of the most notable being PKU.
PKU, or phenylketonuria, is a fascinating example of how knowing the code allows us to completely bypass a disease. It's a recessive genetic condition where a baby is born lacking the specific enzyme needed to digest an amino acid called phenylalanine, which is found in a lot of common foods.
Okay, so they can't digest it.
Right. And if left untreated, this amino acid rapidly builds up in the baby's bloodstream, leading to severe, irreversible brain damage.
But because we screen for it on day one,
We can entirely manage it. The treatment isn't complex gene therapy. It is just a highly controlled, specialized diet, completely free of that amino acid. If the child adheres to the diet, they grow up entirely healthy.
that's amazing.
The genetic mutation is still there, but by altering the environment, in this case the diet, the disease is neutralized.
That is the absolute best case scenario for genetic screening. But science allows us to look much earlier, deep into a pregnancy. Prenatal testing gives parents crucial information, but the methods have vastly different timelines and risks.
They do.
We're looking at chorionic villus sampling, or CVS, and amniocentesis. Now, CVS takes a tiny sample of placental tissue, and doctors can perform this quite early, around 8 to 10 weeks into the pregnancy.
That's right.
Amniocentesis, on the other hand, requires a needle to draw amniotic fluid, and parents usually have to wait until the 16th week of pregnancy to do it. If you can get the genetic information at 8 weeks with CVS, why would a family ever endure the anxiety of waiting 2 whole months for an amniocentesis?
It comes down to an agonizing balancing act between early information, clinical blind spots, and medical risk. CVS provides answers in the first trimester, which is emotionally easier for many families. However, CVS carries a slightly higher risk of triggering a miscarriage compared to amniocentesis.
Okay, that's a big factor.
But more importantly, CVS has a profound biological blind spot. Because it tests the placenta, it runs into an issue with the sex chromosomes. In placental tissue, the paternal X chromosome is naturally inactivated. The genes are literally switched off.
Wait, so if the placenta shuts off the father's X chromosome, you just can't read it?
Exactly. If doctors are specifically looking for a genetic mutation that the fetus might have inherited from the father on the X chromosome, CVS is literally blind to it. It will not show up on the test.
That's a huge limitation.
It is. Amniocentesis, because it tests actual fetal cells floating in the amniotic fluid, provides a much more comprehensive and accurate genetic picture, even though the wait to get to the 16th week can be psychologically grueling for the parents.
And for families who already know they are carriers of a severe condition like CF or hemophilia, the technology has pushed even further to something called pre-implantation genetic diagnosis, or PGD.
Which is incredible technology.
This is where doctors use IVF to create embryos in a lab. When the embryo is just a tiny microscopic cluster of cells, they carefully remove a single cell, sequence its DNA, and identify exactly which embryos carry the devastating mutations. They only implant the healthy ones. They are quite literally editing the disease out of the family's future lineage.
The science is staggering, but the ethical weight of these technologies is equally immense. Genetic counseling has become a vital medical field because interpreting this data is not just a scientific calculation. It forces profoundly personal decisions.
Yeah, I can't imagine the stress.
Think of the emotional toll of a false positive on a prenatal test, where parents might make permanent decisions about a pregnancy based on flawed data. Or consider the heavy burden on parents trying to navigate their deep religious beliefs regarding medical intervention in reproduction.
And there is a societal aspect to this that is deeply psychological. When medical science possesses the tools to screen for and potentially eliminate an ever-growing list of genetic defects, parents often feel an immense unspoken societal pressure to produce a perfect baby.
Indeed. The mere existence of the technology creates an underlying expectation that it must be used, and that any preventable suffering is somehow a failure on the part of the parents. That is an incredibly heavy burden to place on any family.
We have covered a massive amount of ground today. We started at the absolute foundation, watching how a single substituted letter of DNA, a simple swap of an A for a T, can cascade into altered proteins that collapse red blood cells and block arteries.
The butterfly effect of biology.
Truly. We navigated the sheer casino of genetic inheritance, where every child represents an entirely independent coin toss. We looked at how the evolutionary mismatch of the X and Y chromosomes leaves men uniquely vulnerable.
And how a tiny locked doorway causes cystic fibrosis.
Right, that systemic shutdown. And finally, we arrived at the heavy, complex frontiers of genetic screening.
We truly are at a unique point in human history, where we are not just reading our own source code, but learning how to intervene in its expression.
And that leaves us with a final thought to ponder. If science continues to advance, allowing us to aggressively proofread our genetic manuscript and eventually edit out all our perceived genetic flaws, well, where do we draw the line between preventing devastating suffering and erasing the very natural variations that have driven human evolution since the beginning? Is the pursuit of a perfect genome actually a dangerous myth? Thank you for joining us on this deep dive. It was custom tailored just for you to help you decode the incredible complexity of what makes you human. Until next time, stay curious.